AVINASH VS, Assistant Professor and Head Department of Biotechnology. Today let's see the overview of the biomolecules as a part of our syllabus. Okay, so this is about the all biomolecules which are required for our day-to-day life, saying proteins, carbohydrate, lipids, nucleic acids, etc. Okay, so this includes vitamins also. Okay. So I'll be dealing with only the introduction and the introductory concepts of the carbohydrates. Okay, so carbohydrates, as the name suggests, it's a class of a biomolecules which is having an empirical formula CnH2ON. Okay, so this is an empirical formula. We can just add N by 1, 2, 3 and we can get the structures of the biomolecules like carbohydrates. The minimum carbon number which can be added here to be referred as carbohydrates are C3. Okay, so C3H6O3. So this is a basic carbohydrate which we can say, and we call them as triose. So we can add a 4 carbon, we can say it as tetrose, we can add 5 carbon, we can call it as pentose, we can add 6 carbon, and we call them as hexoses. And hexoses are usually the most abundantly seen and which we can equate when we say about carbohydrates. So we can generalize glucose, which is a 6 carbon compound having the formula C6H12O6. Now we can see that it is following the empirical formula CnH2ON. So where carbon is replaced with 6, hydrogen by 12 and oxygen N is with 6. So this is the best known carbohydrates and we say this as glucose, and we can use this as a reference and we can study the other carbohydrates, saying the disaccharides or polysaccharides or the other forms. Okay, so here we can go with the empirical formula or with the structures. So when we say about the structure, we can say the projections. So Emil Fischer in 1891, he proposed this structure where which we had written like C6H12O6, which was only the structural formula, the chemical formula, but we have to write the structure also, a three-dimensional structure where we can say where exactly the CHO will be, where exactly the CH2OH will be, what will be present at the 5th carbon, what will be present at the 4th carbon. So to understand these, Emil Fischer in 1891, he proposed the structure and we call it as the Fischer projections. And later that was been improved by writing the three-dimensional structure or the components which are in three-dimensional, which we can reduce it to two-dimensional, and that was given by Haworth. Okay, so Haworth projection and Emil Fischer's projection help us to understand what is the actual structure we have for carbohydrates. So that's the basic importance what we should be aware when we say about carbohydrates. So here we can say about the structure, the Emil Fischer structure of a ribose, which can be the 5 carbon sugar, which we know as the RNA or the DNA, which is the backbone of those two nucleic acids, which we will be studying at the later classes. Okay, or the galactose, which is the epimer of the glucose, where the variation will be at the C4 positions. Okay, or we can even say the components like fructose. So fructose, which we say as the fruit sugar. So all known fruits which have that sweet taste, that's due to the fructose content what it is having. Okay, so let's see how we can classify these carbohydrates. So first we have to define, saying that it's an empirical formula having CnH2ON. Now we have to say what is the classification which we have. So carbohydrates, they have many classifications, but only 2 classification, that's the accepted classification, says the presence of the monosaccharides and polysaccharides. So this is the only accepted classification we have for carbohydrates. Now you may ask, who to decide this? So we have an International Union of Pure and Applied Chemistry which says this is the accepted classification by referring to the various sources, and says this can be used as a material so that we can classify the carbohydrates as the monosaccharides and polysaccharides. So whatever comes next to this will be the subdivision, saying that monosaccharides can be subdivided into aldoses and ketoses. So here OSE just refers to the sugars. Now wherever you can come across with the term OSE, you can just equate that to sugar. Raffinose, arabinose. So they are all sugars but with the higher structures. That means they have the higher molecular weight, or they may have the carbon number at the higher stages. Okay, so minimum carbon number, it's been seen that it is 3. So we can get around C22 or so can also be established. Okay, so OSE, if it is ending, it is with respect to sugars. So monosaccharides, they have been subdivided as aldoses and ketoses. Aldose is like glucose and ketose the fructose. So I'll go into this details in future classes. So polysaccharides as the oligosaccharides and disaccharides. So these are the classifications we have. So first, it is the carbohydrate, which is a major component of the other biomolecules, which we can say like protein, lipids, nucleic acids. So all these will be required for the day-to-day life. So we need carbohydrates for energy supplements, or we need proteins as an energy supplement. We need lipids as an energy supplement. We need nucleic acids, which has to be present in the diet, and that can act as an energy supplement. Okay, so likewise we have many biomolecules which we require to be acted as the energy supplement, of which carbohydrate is most important. To be frank, it is the protein which comes first, later is the carbohydrate, but we say the maximum share in our diet will be carbohydrate, then will be the protein. So we say we take 1 cup of rice and 2 spoons of dal. So that says about the importance of carbohydrate. So rice or disaccharides like lactose, a component of milk sugar, or maltose as a product of the polysaccharide digestion. So all those will have a major share with carbohydrates, and we require them. Okay, so this is the classification we have. It's monosaccharide and polysaccharide. Whatever comes next will be the subdivision, which is made to ease ourselves to understand their structure. Okay. So monosaccharides will have aldoses and ketoses. They are the subdivisions. Aldose will have the examples like glucose, and here it starts with CHO, aldehydic derivatives. So anything which starts with this is an aldose. Okay. And fructose will start with CH2OH. So I'll write the structure at the later stage so that we can say what is an aldose and what is the ketose and how to differentiate it. Okay, so oligosaccharides are those sugars which have more than 3 sugar units. So anything which have more than 3 sugar units will be known as oligosaccharides, and those sugars which have 2 sugar units will be known as disaccharides. Okay, so we can give the examples for these also, but let's see at the later stages. Now let's see the structure of an aldose. So it starts with CHO. I'll be writing the Fischer structure. So CHO will be the first, and I'll be writing the structure of a glucose. So it is an hexose having the 6 carbon number. Now you can count 1, 2, 3, 4, 5. and 6. So 6, in whatever the sugar we write, it ends with the CH2OH. Okay, so this is an structure, what we can say, like the Fischer structure of glucose. It starts with CHO. Later we have to write where the hydroxyl groups are present. So whenever I write the arrow or the line that represents the location where hydroxyl groups are present, and opposite to it will be the hydrogen. So let's fill this first. 1, 2, 3. Okay, so OH, H, OH, H, OH, H. So this will be continued, and we can say this as D-glucose. Now I'll be saying about the new concept or the components which they. We have seen here, I had written an uppercase D or capital D. Okay, so we can even write this. So what this uppercase D and lowercase d notes? An uppercase D says about the orientation of the hydroxyl groups which is at the penultimate carbon atom. Now we can see I had written the carbons of 6. So penultimate is the 5th carbon atom. So if this hydroxyl group, if it is on the right side, we can say this as an uppercase D. If the same hydroxyl group, if it was at left, then we would have said it as L, uppercase L. Don't get confused with the dextrorotatory and levorotatory, which I will be saying now. Okay, so lowercase d is the property of a carbohydrate to turn the plain polarized light to right. If you make a solution of this, we know that glucose can readily dissolve in water, so we can add 2 spoons or 3 spoons of sugar, stir it well, we'll get a solution. So when the solution of this is being passed through a monochromatic light, that means a light with a single beam, then it deviates to right because we had taken the glucose which is known as the dextrorotatory. Now we have on the other hand where the hydroxyl group at this point can turn to at the left, and we say it as levorotatory. That means the plain polarized light turns towards the left. So if the light turns to right, it will be dextrorotatory. If it turns to left, it will be levorotatory. These are the properties of the carbohydrates. Okay, certain proteins also possess this property, but as we are dealing with the carbohydrate, so let's confine ourselves to the class of carbohydrates and let's see what are the components which we can say here. Okay, so this is about the structure of glucose and an example coming under actose. Now we have to write the structure of a ketose. So ketose, as we say, the first carbon number will be having CH2OH. I'll just make the arrangements here, CH2OH. Rest all the things remain same, and now we can say this as D-fructose. Again, we use the term uppercase D and fructose. Okay. Similarly, we can write the lowercase d also. Uppercase D is saying about the orientation of the hydroxyl group which is towards the right side of the penultimate carbon atom, and lowercase d says about the levorotatory property or dextrorotatory property. Now I think it's clear. Now we can proceed to the oligosaccharides and disaccharides. So let's see the examples which we have here, then we can write the structures. Structures will be like this. So if we can say about the carbon number, we can write the structure. So the structure or the pattern what I had written here is the Fischer structure. So this was proposed by Fischer. Now if you ask how to write the Haworth projection, it's an open chain structure. Haworth projection will be closed ring structure. I'll just write the Haworth projection of the carbohydrate. It will be like this. I'll just rub all this. Okay. Now I had written the structure of the Haworth ring structure. Earlier it was the Fisher, now it is the Haworth. Now what is these lines? These are the locations again where the hydroxyl groups are present, and opposite to it will be the hydrogen. A hydroxyl and the hydrogen. So this you can complete. So this is the projection which was proposed by Haworth. So we can write the projections of this Haworth projections and Fisher's projection for all the class of carbohydrates. We can write this structure for the disaccharides. We can write the structures for the polysaccharides. We can write the structure even for the oligosaccharides. But in our syllabus, we don't go into the details of the oligosaccharides. We confine ourselves to the monosaccharides, saying the structure, the examples of the ribose, D-ribose, which is a pentose sugar, or glucose, which is a hexose, a 6-carbon sugar, galactose, again a 6-carbon sugar, and we have a relation between a glucose and a galactose. So what is that relation? It is an epimer. So epimer is a condition of the stereogenicity where the variation will be at a specific position, a specific carbon number. So we can see the variation at C2 position. Glucose and mannose is an example for the epimers at C2 positions, and C4 positions can be seen changed in glucose and galactose. Okay. So this is a stereogenic property which we can say in carbohydrates, but not seen in any of the other biomolecules. Okay, so we can write the structures, these structures for the polysaccharides. We can write this for monosaccharides. We can even write for the disaccharides. Now let's see the examples which we can quote under the disaccharides. So examples which we can say is maltose, lactose and sucrose. So all these are the best known or very used commonly in our day-to-day life. Okay. One is not present naturally but seen as a part of digestion, that is maltose. So we consume polysaccharide in our diet like rice or glycogen, a part of the animal sugar. So rice, a plant sugar, starch, that's present as a polysaccharide, or we can say about glycogen, which is a part of the animal sugar. So all these are the examples of the polysaccharides, and when we eat this has to get digested, and in the way of its digestion at the secondary site of digestion, that is stomach in our body, it gets broken down into amylose, the products, and that gives the maltose. So maltose have 2 glucose units. Okay. Next disaccharide which we can say is lactose. Lactose is best known as we have that to be present in our day-to-day life. It is the milk sugar. So every day we used to drink milk, and 1 cup or 2 cup, whatever it may be. So we get the sugar or the lactose from those milk. And finally the sucrose, which is known as the sugar of commerce, the cane sugar or the beet sugar. They are the sources by which we can get these disaccharides, and they have a specific role to play. Okay, so if we come to the class of the polysaccharides, so we can say the rice, which is having the starch, and this starch is made up of 2 subunits like amylose and amylopectin. Okay, so one is a linear chain of 25 to 30 carbohydrate residues, and over that we can see branching at 7 to 12 branching points. Okay, so it's like this, a linear carbohydrates, and after 7 glucose units we can see a branch. Again it continues. Okay, so this linear chain is known as amylose, and the branching point is known as amylopectin. So both reacts positively with the iodine solution and gives blue color. Okay. So this is also a property exhibited. That means whenever we can test the presence of these polysaccharides, we usually go with the use of the iodine test. Iodine test to detect the presence of starch. Okay, so we can go with the usage of potato. We can take the potato tuber, peel its outer skin, just add few drops of iodine, you can see the color. That's due to the presence of the starch. Okay, and amylose will also react with that starch and forms the starch iodine complex and gives the blue color. Okay, so similarly we can say about the glycogen. Glycogen, which is the animal source of the carbohydrate, which we can say as animal starch. Okay, so plant starch, we have that as the rice, and an equated version of that which we can see in animals is the glycogen, which we can say as animal starch. Now both have the ability to react with the iodine solution. So I said when we add the iodine solution to rice and all, it gives blue color, but when we add this to animal starch, that is glycogen, it gives violet color. So violet color is given by the presence of this. And only difference here what we have here is the branching point. Earlier we had like 7 to 12 in case of the starch, but in case of glycogen we can see the branching point at every 25 to 30 sugar units. This is the usual way which we use to define the carbon number. So here I am using the term sugar unit. That means I am referring to glucose. So that is the context which we should take whenever we use the term sugar units. 12 sugar units, 13 sugar units or 20 sugar units means that it has 20 or so such numbered glucose residues. So this is with respect to the sugar units which it is having and the concepts called polysaccharide. Later we should say about the other properties, saying reducing and non-reducing property. Okay, so all carbohydrate doesn't have this ability. That means any sugar solution which can readily react with benedicts, phenol's and toluenes. So when it reacts positively with benedict, phenol's and toluenes, we can say such sugars as reducing. Any sugar which doesn't react with these positively will be known as non-reducing sugars. Okay. So a reducing sugar reacts positively with benedict, phenol's, toluenes, and a sugar which doesn't react positively with benedict, phenol, toluenes are known as non-reducing sugars. Now, after understanding of all these, we should say what is the importance we have with respect to biological entity. That means the bioimportance of carbohydrates. Why do we need this? And what is the amount of carbohydrates that has to be present in our body or present in our diet? Okay. So we say that in our mornings we should have enough constituents or enough supply of the monosaccharides, glucose, or we can go even with the specific disaccharides like lactose. So that's the usual practice which we follow. Usually we start our day with a cup of milk or a cup of coffee which has the milk. So milk sugar is lactose. That is the usual component which is used, and that provides some amount of energy requirement for day-to-day activity. But that is not the permanent one. So we should switch to the higher versions of the diet, saying we should have a breakfast. And a breakfast will have again the disaccharides and always it should follow or supplement with the polysaccharides. So even in the lunch, we need a major share of the polysaccharides, and even at the dinner, we require a major share of the disaccharides and polysaccharides. So that is one important requirement for maintaining body's activity. Now, what is the other requirement? Why do we need these other than the nutritional supplement and all? Some cells require a constant supply of energy. Brain and nerve cells, they require a constant supply. It's not like that I had a breakfast at 9 so that I can skip my meal, I can have my dinner by 7 so that I'm advancing it. No, we need the constant supply of energy, and that's given by the diets what we consume, and a major share, around 80% of this, will be taken by brain cells, and next will be nerve cells. Later, all the available energy will be utilized by the other cells of our body to maintain their nutritional requirements. So that is the importance. So we should not skip our meal, and we should not think that I can advance my meal or advance my diet plan, or anything. Such things should not be followed, because we have a very rigid pattern saying that all the bodies which requires an instant supply of energy will be in queue, and they will be waiting for the energy requirements, and if we don't provide it, we know what will happen. Okay. So this is the importance we have for the carbohydrates. Next, we can say about the other nutritional importance like proteins. So G.J. Muller in 1891, he proposed the structure, or he said about proteins, and he said this, he used the term proteose, that means to occupying the prime place of importance, which comes at first. So that is the most required nutrient which has to be supplemented to our body. Why do we need proteins? Proteins are required for various bodies function. We need we need protein for our hair, we need proteins for our nails, we need protein for the body's shape. So all these are given by the proteins. So that's the next component which we have in our pattern. First is the carbohydrate, which says about all these things. Next will be the proteins, which says about the primary structure, secondary structure, quaternary structure, and other forms. So I'll just give an introduction for the primary proteins, and we can see it in the later class. Like so insulin, it's a primary protein. Some says it as hormone. Yes, it is a hormone, but it is also a protein. That's because it is having the monomeric units like amino acids. As we had glucose as the monomeric units for larger class of polysaccharides, saying starch, glycogen, or chondroitin sulfate, which we can say it in any form, all will have the monomeric units as the carbohydrate or glucose. So here the monomeric units we can have is amino acids. Okay, so this constitutes, this can be treated as the monomeric unit, and a large class of this makes the specific proteins. Insulin is one such protein which is having a branched structure, having A chain and B chain, A chain and B chain, A chain having 21, B chain having 30 amino acid residues, and it has 4 disulfide bridges. and here. So this is the structure which we have for insulin. Now, what is the role of this insulin? It's a hormone which is used to control the blood sugar level. So we can equate this to the earlier class of carbohydrates. So carbohydrates, if it is being overeaten or if we consume the highest amount of starch or higher amounts of the carbohydrates in our body, and if we don't exercise, we'll develop that as specific forms. That's a new component which we will study at later class. So that has to be lowered. We should not accumulate, or we should not give these components to settle in our body. It should be completely digested as well as it should be completely absorbed. So when it is not being absorbed, it remains in blood, which is the universal transporter which we have in our body. So which transports the oxygen, and which is having a carrier like hemoglobin. The condition will be oxyhemoglobin, where the hemoglobin will be rich with oxygen. It travels to parts of body, and wherever the cells require oxygen, it gives the oxygen from the hemoglobin and takes out the carbon dioxide, and reaches the heart for purification as decarboxy or carboxyhemoglobin. Deoxyhemoglobin or carboxyhemoglobin is the condition where the carbon dioxide content is increased. So that's the usual practice. So blood should contain only either oxygen or carbon dioxide. When the blood have the traces of sugar, so we usually say the condition as diabetic. When a person is said to be diabetic, he will be having the levels which is being not absorbed by the body to be present in the blood. So he'll be having the sugar, he'll be eating the normal diet, he may not be aware or he may be aware, whatever may be the condition. So he will be having the carbohydrate as a part of the diet, probably like lactose, a milk or a coffee which he consumes. Okay, so that will reach the body and it follows the process of digestion. In this event, when it fails to absorb at the site of small intestine all the components which has been completely degraded or digested, that gets absorbed. So when it absorbs, it will be in the form of carbon, hydrogen, oxygen, nitrogen, phosphorus, CHONP. So these can be absorbed by the cells where it requires carbon. If cells require oxygen, it can be taken. If the cells require nitrogen, it can be taken. So this is the final part of the digestion. It leads to absorption. But when the individual fails to completely digest carbohydrates like glucose, then the levels of glucose mixes with the blood and it starts to travel parts of the body. Now it's an early signs of diabetic. Okay, so if we test, taking the blood sample and subjecting it to specific test, Benedict test. Again, it's a test to see the presence of the sugar. If there is a change in the color, we can say the condition as diabetic. Based on the color intensity, we can say whether they are in a pre-diabetic condition or in a post-diabetic conditions. Okay, so insulin in such patients will not be secreted in the usual quantity. So this is the relation which we have between carbohydrates and proteins. Proteins are required to utilize or completely absorb the carbohydrates which was taken in a diet. Okay, so glucose is a part of a carbohydrate and it's a monosaccharide. Everything is known. This has to be completely absorbed in our body. Okay, so when it is not being completely absorbed, if the traces are present, then that has to be taken with the presence of insulin. So that will lower the levels of carbohydrate. So when this condition is also not seen, then the condition is diabetic. Okay. So this is the relation which we have for the proteins and carbohydrates. And next we have oxytocin, which is known to be an octapeptide. Insulin, we said 30 amino acid residues. Oxytocin will have 8 amino acid residues. And we usually have a practice to read those amino acid. I'll just write this structure, not writing the structure of oxytocin. Glycyl, valyl, isoleucyl. That should be the way of reading the amino acids. We usually read this as glycine, valine, isoleucine, but these readings, if it is in a single terms, that means when it is not in a peptide form, if it is in single, we can say it as glycine, we can say it as valine, we can say it as isoleucine or so. But when it is in a structure, when it is in a combination, or when it is in a peptide form, we should use the term like glycyl, valyl, isoleucyl, and it continues. Now, what is the specialty of this oxytocin? It's a hormone which is used, or which is known as the female hormone, very usually seen in females, and especially in lactating mothers and breastfeeding mothers. So it helps in those products. So we can give the other examples also for these proteins, saying keratin, collagen, or hemoglobin, which we were saying like it carries oxygen and says as oxyhemoglobin, or takes carbon dioxide and we say it as deoxy or carboxyhemoglobin. Okay, so all these are the examples of proteins. But it can be a primary protein, it can be a secondary protein, a tertiary protein, a quaternary protein like collagen, a triple helical structure. Okay, so all these are the variations which we can find in our body, and proteins are required for structural organization, but carbohydrates are used for maintaining body's function. We don't say that proteins are not required for body's function. They do require, but a major share is taken by these. Okay, so this is about the way of the orientations which we have for the proteins. Next, we can say about the secondary structure or quaternary structure, and the subclassifications which are present here. Collagen and alpha helical pattern, or keratin, which is seen in now this is about the subdivisions which we have in the proteins. I said about the primary protein, they will be linear without any branches. Branches will be only of the disulfide bridges, and that's due to, or it's required for maintaining the structure. Now if I say about the secondary structure, I can give the example as the alpha helical pattern and beta plated sheets. Now what are these? This is the helical pattern which is seen, yes, DNA, the genetic material of us or any organism, prokaryotes, eukaryotes, or let's not go into those details. So DNA is a helical molecule. So helical pattern, and we are using the term alpha, and here alpha refers to the first component or the first type of secondary structure that is being seen. Okay, so alpha helical pattern. The next type what has been noticed or identified is the beta pleated sheets. Now how to write this? It is just the asbestos sheets what we know. It's the best example for the pleated sheets. Pleated refers to the pile one above the other. Okay, and here we can see all these are at the same distances. Okay, the ups and downs what we can say in the local terms. So that's seen in running in the same direction, or we can even see those which runs in anti-parallel. But forget about those. It is the second way of writing the structure. It is beta pleated sheets. That means sheets which has been piled one above the other. Okay, so alpha helical pattern and beta pleated sheets. So these have been identified as a part is the secondary structure of proteins, and keratin and collagen are the examples which we have for the secondary structure of proteins. So if we go for the quaternary structure, we can say about the hemoglobin, myoglobin and others, where there will be no such diversions, but it will be having many disulfide bridges, and it can't be in either the linear fashion or it can't be in any branched fashion. So it has to be having the twist and turns like this, and amino acids can be linked. Okay, so wherever there are branches, we can see the presence of the disulfide bridges. So this is about the tertiary. Quaternary will be having many such. Okay, so this is about the protein and their structural organization. Again, we have to say about the biological importance. What is the biological importance we have for proteins? Okay. So why do we require proteins? The simple answer: we need hair, we need nails, we need a straight body. We don't want to be lean or bending, so we should walk, stay fast, those, and all that's given by proteins. So that's the first in case of the biomolecules. Bio refers to living molecules. All those which are living, these are the components which are needed for them. What are the components which they need? They are proteins. Then comes carbohydrates. Then comes the lipids. And finally, nucleic acids. So in this class, we said about the proteins. We said about the classification of it, saying primary proteins, which doesn't have any branches, it will be linear arranged. We said about the secondary structure of protein, where there will be branches like alpha helic or beta pleated sheets. Fine. Next, we said about the tertiary proteins, and we said even about quaternary proteins. Okay. Next, we said about the carbohydrates. Carbohydrates like the monosaccharides. I'm just saying about the classification. Monosaccharides and polysaccharides. And they are subdivided as aldose. Monosaccharides are subdivided as aldoses and ketoses. And in case of the polysaccharides, it's further subdivided as the disaccharides and oligosaccharides. So these classifications are based on the carbon number. If there is only a presence of 2 sugar units, it is a disaccharide. I said an example of maltose, which is having 2 sugar units or glucose units. Lactose, which is having glucose and galactose. Likewise, okay, so they are having only two sugar units, so they are referred as disaccharides. If they are having more than two sugar units, they are referred as oligosaccharides. And if they are having only one sugar unit, they are referred as monosaccharides. And monosaccharides can be further subdivided as aldoses and ketoses. Aldose in examples like glucose, and anything with CHO to start, they are all aldoses. And ketoses, CH2OH. Okay, so that's about the structure or the way by which we can have a classification for the carbohydrates. Then comes the lipids, and finally the nucleic acids. So in our diet pattern, if you can see your an afternoon diet, you will be having this, but some people will try to skip this, but that's not advisable. So we should not skip any of these. To ask a question, how we can get nucleic acid in our diet? Don't we do we have that in any natural form? Can we get that from leaves? Can we get that from the vegetables? Or can we get this from any of the animal foods? Yes, but we don't require that in essential forms. They do require and they should be present in our diet, but not as the class of proteins, carbohydrates, and lipids. Okay. Lipids, the examples is oil and ghee, butter, coconut oil. Some people usually use this as a part of their diet pattern. Okay, coconut oil and arachidonic acid. That's also an example of a lipid. Okay, and the peanut oil, groundnut oil, sunflower oil, sesame oil. So we can give n number of list, and all those can be said as the lipids. No. We said about the monomeric units. Let me say about the monomeric units of lipids. So if I say proteins, the monomeric units will be amino acids. Now if I say carbohydrates, the monomeric units will be glucose. Now if I say lipids, the monomeric units will be fats and glycerol. So these are the two monomeric units which will be there, which will be added in volumes so as to make the complex biomolecule called lipids. Now later we can say about the examples like groundnut oil, sunflower oil or coconut oil, butter, ghee, vanaspati, XYZ. Anything what we can say will be having fats and glycerol. So these are considered to be the monomeric units, and they help in maintaining again the body's function, or it gives the stability. Now I am asking a question, how lipids helps? Lipids play a major role in maintaining our body's function by providing a blood-brain barrier which is there in the brain. So if we don't have that, if we don't have a blood-brain barrier which is having a major share with the lipids, then we would not have been the ability to stand or to hold this, to write, nothing. So with this barrier, we are having the ability to control some of these things, like I can hold this, or I can hold the pen, I can write, I can speak to you, or I can stand still. All those are due to those factors. Okay, so this is about the lipids. Finally, with the nucleic acids, we can say two forms. One is DNA, another is RNA. Deoxyribonucleic acid, again having a association with carbohydrate. deoxyribose. Ribose is the sugar. Sugar means carbohydrate. Okay. RNA, ribose nucleic acid. Again the sugar is ribose. Number of carbohydrate having the carbon number 5. Okay. So I'll just write this structure, Fisher structure for ribose. CHO, it's an aldose. It's a 5 carbon number, 1, 2, 3, 4 and 5. Last is usually CH2OH, 1, 2, 3. Now you can say this is as D ribose. Okay, so place has to be filled with hydroxyl groups. Opposite, yes, hydrogen. So this is the structure of D. Hydroxyl is towards right, and D. If we can get this solution, we can see, we can usually represent this, not a problem, D ribose. Similarly, we can have L ribose also. Okay, I said proteins also have this property, like L-alanine, D-alanine. Now you can see I have not written the structure, so don't go with the searching of the hydroxyl group whether it is towards right, left. Now that's not the concept. L and D is with respect to the COOH group. So structure, the monomeric unit of amino acid will have a centrally placed carbon atom. To right side will be COOH carboxyl group and N-terminal NH2, an upper hydrogen and lower replaceable R group. Now this is the example for D. Okay, COOH, D, whatever may be. If I just replace this R with H, this will be the most simplest amino acid, glycine, where R group is just replaced with hydrogen. So I can replace this hydrogen group with the aromatic groups. We have aromatic amino acids also, tryptophan, and so we have aliphatic amino acids. Let's not go into those details. I'm just saying L-alanine and D-alanine. This is just example of an amino acid, and here we can see in nature both the types. But we may require only one form for our diet, and we classify them as the essential amino acids, and some are the non-essential amino acids. That means amino acids which is required to be present as a part of the diet will be essential. Any amino acid which is required to be present as a part of a body's food diet pattern, such amino acids will be known as essential. It means they have to be present in a diet. And we have the ability to produce some amino acid as a part of the metabolic reactions. I have not said any of those metabolic reactions. In next semesters we can study that. Okay, so as a part of the metabolic reactions, we can see some amino acids can be formed. So such amino acids we can say as non-essential. Essential amino acids are those which is required to be present as a part of a diet. Non-essential, it's not required to be present and it can be synthesized in a body. Okay. We have the ability, as humans, we have the ability to synthesize 50% of the amino acids what we have listed or stated so far. So we have listed around 20 amino acids so far, and 10 amino acids are referred to be as essential. That means they have to be present in our diet, and we have the non-essential where it is not required to be present, and we say it as non-essential. So let's write the essential: P, V, T, T, I, M, H, A, L, L. So these are the amino acids: proline, valine, tyrosine, tryptophan, isoleucine, methionine, histidine, arginine, leucine, lysine. So these are the amino acids which we call as essential. So these has to be present in a diet. It can be in a supplement or it can be in a diet. Now what is supplement? The supplements are the food which is usually used as a replacement for a diet. So sometimes we can't have the breakfast, so we usually go with 1 cup of milk by adding 2 spoons of Horlicks XYZ. So they are the supplements. So they will have these amino acids. So you can just check the pack of it. At least some will be present. We say that all has to be present, but there is a saying like as it is a supplement, we don't rely more on supplement, we rely more on the diet. So we depend more on our food than supplement. We take only 2 spoons to be added either in milk or some even add that to water and consume it, but our diet will not be of 2 spoons or 3 spoons. So we can see or we can understand what's the magnitude we have for the diet and for the supplement. But this supplement will have any of these essential amino acids, and the left out like glycine, which I have not stated, can be found, as it's a simple amino acid where R group is just replaced with the hydrogen atom. We can get that. Okay, so we can list the other amino acids also, and we can say that as non-essential amino acids. Okay, so this is about the essential, non-essential amino acids which comes in the class of proteins. So I think I can conclude this. Thank you.